Okay, we hope you had an enjoyable evening last night at the dinner upstairs. I think, and those that I think got the awards last night, congratulations, and I think they all got a buzz from it. So yeah, it was a good night. I do have to. And apologies again for the people who've probably heard this for about 5 times. We do have to go over the health and safety instructions. So look, if the alarms go and we have to bail, the exits that way, and I think that's the main one there. There's one out that way. So use that one there. And the assembly area is out in the car park. There's a grass park out there, so assembly out there. You know what to do in an earthquake. We are in an active earthquake zone, so under the tables, and stay there until everything settles down, and then leave the building through the exits. And we do, the instructions are if there is an earthquake and there's a tsunami warning, we do have to go up one floor because we're not high enough here, so which is a bit scary. Okay, any medical issues, obviously normal 111 or get assistance from the Te Papa people here. Okay, just a few announcements before we kick off. There are pamphlets and reports that a number of the presenters have left, and they're on the table out there. So if there's any, and there are some of these science reports and other things there, if anyone wants copies of those. And I think there are some spare copies, a few spare copies of the presentations there as well, if you do want to take those. Right, don't forget there are some posters out there that a number of people have prepared, and the people who've prepared those will be around those posters at morning tea and lunch breaks. So yeah, have a look at those and talk to the people who've done the work to bring those along here. In the program, we have got the woodlot harvesting calculator tool will be demonstrated. That won't be demonstrated, so that won't be happening. We had to pull that out of the program. We are going to be using the app again, the Poll Everywhere, and the instructions on how to get onto that are in your bag. You just go to the browser on your laptop or your cell phone or tablet and post questions on there. If you need any help with that, where's Max? Max is over in the back corner there. Max from Scion is helping, and he's running that, so any help, see Max. We will have to keep rigidly to time today. So we've got a timekeeper with a bell, and the timekeeper will give a ring at 5 minutes and 1 minute. Marco, is that? Yeah, they'll know. They'll know? Okay. As long as they know it's 5 minutes and 1 minute, and then if you don't stop talking, then someone big and will come up here and take you off the stage. And so that we'll be doing that, and we've got a chairman. Chairs have been volunteered for each of the 4 sessions. So I've got Mike Baker from Hancock for this first session. Dean Woodahira for the second session, although Dean texts me, say his flight's been cancelled or delayed or something out of Rotorua, so he's not sure when he'll get here. So we'll just keep a watch on that one. Angus Gordon, Angus from the Farm Forestry Association will be chairing the first session after lunch, and Jason Syme the final session. So I think that's all we need to say, and I'll hand over to Mike Baker from Hancock, who will be chairing this first session. So, thanks, Mike.
Good morning, everybody. You're welcome to this first session on day 2. This is titled Protecting Our Future. It's focused on forest health research, and particularly pathogens that affect our commercial species, and particularly, and also, in one example, our native species. There's 2 parts to this session. The first one's around the Healthy Trees and Healthy Future program and the Nettle Disease Research Program, and secondly, there's another part on the potential for biocontrol of pathogens. In regards to the Healthy Trees, Healthy Future program, it's been a 6-year program with significant funding from MBIE and also from the Levy and other parties. This program has been aimed to improve our knowledge around Phytophthora diseases, which is responsible for red needlecast in radiata pine, kodu dieback, and also losses in our horticultural crops such as apples. The program has been cross-sector, led by Scion, involves other parties such as Plant and Food and Landcare Research. In regards to the needle disease research program, this has been primarily focused on, again, red needlecast. It's aimed at understanding the extent of the disease and its environmental conditions that favour the development of the disease, its growth impact, and also potential control options. There's also 3 other well-known diseases involved in that program: Dothostroma, Cyclonusma, and physiological needle blight. In regards to the second part of the session and the potential for control of biocontrol of pathogens, this has been led by the Bioprotection Research Centre at Lincoln, and the aim of this one has been to identify beneficial organisms that will improve the health and productivity of not only radiata pine but also other species that's been looked at in cypresses and Douglas fir. And Travis Glare, who's the director and from the Bioprotection Centre, will be speaking, as well as Helen Whelan, who's leading the Trichoderma Forestry Research Program there. And I should also mention that there's apologies because their profiles have been omitted from the proceedings. But firstly, I would like to welcome the Scion, who's talking about the Healthy Trees and Healthy Future and needle disease programs. We have Lindsay Ballman, Alan Tan, Natalie Graham, and Rebecca McDougall speaking. And Lindsay, who leads the forest protection team at Scion, will be starting first. So Lindsay, welcome.
Thank you, Mike. Good morning, everyone. Before I start on Healthy Trees, Healthy Future, I just want to give a little bit of a history lesson, I guess, on some of how we've handled new pathogens that have arrived in the country previously. So the first one is Nectaria fluke canker. It was confirmed in 2003. It really did have us scratching our heads. The pathogen was noted on spruce and fir trees in the northern hemisphere. It was noted as a wound invader. It wasn't a serious pathogen at all. It arrived in New Zealand in the southern hemisphere for the first time on radiata and caused considerable damage and really was serious concern to forest growers in the southern South Island over that period. Caused severe malformation of the stem, and you can see by that picture on the right there, that's an extreme example, but it really was serious. But through research, we came up with findings that allowed forest growers to alter their silvicultural regimes, and now the pathogen really isn't causing much damage at all. It's a curiosity disease, as Peter Oliver aptly describes. So, it took about 6 years to really control and manage that disease. Dothostroma needle blight, it arrived in 1962, it was confirmed in 1964, causes severe defoliation of pine trees, as I'm sure most of you know. By 1966, we had an operational control method for that, and we were routinely spraying trees with copper. Breeding and spray applications research has certainly helped manage that disease in recent times, and we've gone from spraying at 50 litres a hectare down to 5 litres and sometimes 3 litres a hectare. So great efficiencies made, and another success story. Red needlecast, that's a slightly different story. It was first noted in 2005. This is a forest on the East Cape of the North Island, causing medium severe defoliation. When I looked at this stand then, I thought there was something unusual happening. We took lots and lots of samples, didn't isolate anything new in terms of pathogens. So we just put it down to some odd year, something's funny, but it'll come right. 2008, that's that same stand that I photographed previously. Severe defoliation. We took one look at it, Margaret Dick and I, and said, this is something new, this is something different from what we've seen. Went back to Rotorua the following day. MPI, MAF in those days, and the forest owners were readied, and by Monday we had started an investigation response, so it can happen that quickly. We hadn't really seen, as I say, anything like that before. Just shows you the two different pitches. So when it was confirmed in 2008, it was confirmed as a Phytophthora. The Phytophthora was new to science, something that would never have appeared on any risk list or anything like that. The trade implications were of greater concern than the productivity loss. Forest owners were concerned that China might ban our log imports because of the ability of our logs to carry this pathogen over there and establish over there. So really for the first 3 or 4 years, we determined the risk to trade and through research determined that this pathogen wasn't a risk to our trading partners. And we published that, we presented results in a conference and then published those results. So that issue was mitigated. Then we focused on how to control the disease and increase productivity and avoid productivity loss from that. As Mike said, there's 4 foliage diseases on radiata pine in New Zealand: Dothystroma needle blight, Cyclonesma needle cast. We call it Phytophthora needle blight now, because we know that blight is caused by a Phytophthora, and red needle cast. So what do we do to control these diseases? This is where the Healthy Trees, Healthy Future program comes in. And this is all about Phytophthora diseases and trying to future-proof our forests from the threat of Phytophthora diseases. And really what we tried to do was inoculate 3 different hosts, pine, apple and kauri, with a range of Phytophthora, and look at the responses that those inoculations elicited, and from that we could understand and gain knowledge about how the tree could defend itself from those Phytophthora threats. That was a pretty lofty aim at the start of the program. We didn't get there in terms of doing all the crosses between all the Phytophtheras, all 3 hosts, and roots and foliage, but we got some of the way, and I think the program has been successful despite not meeting those goals. So I'll go into the first part of the program, and that's looking at epidemiology of Phytophthora pluvialis and how it causes red needle cast, how they have this pathogen has the ability to cause epidemics. And what we've found is that it's polycyclic, it causes multiple disease cycles per year, and that can happen really, really, really quickly. The chart on the left can show that disease symptoms can happen 5 days after the pathogen actually infects foliage. That's really, really quick. Second infection can occur in about 20 days. So from that you can understand that this disease can build up really, really quickly, and I've seen that in the field where stands have gone from completely green to pretty much red within a month. So that's what we're sort of battling against. That's for on radiata. On Douglas fir, we did studies where we matched climatic conditions in Oregon, where Phytophthora pluvialis comes from, and conditions in New Zealand. And you can see from that, probably can't see from where you are, but the conditions, some of the conditions match quite closely in terms of rainfall and temperature. What we've found, interestingly, is that in New Zealand, the amount of pathogen in the foliage is significantly higher than it is in Oregon. Maybe in Oregon, because the pathogen's native to Oregon, that the hosts have, evolved with it, and they have the ability to fight off the pathogen, and they're sort of in balance. But in New Zealand, because Phytophthora pluvialis is a new introduction, Douglas fir, maybe we've got a narrower genetic base. The pathogen has much greater ability to infect hosts than in Oregon. Again, so just repeating what I've said really, that there's a lower prevalence of Phytophthora pluvialis in the Pacific Northwest than there is in the South Island of New Zealand. And mild winter temperatures appears to be the key driver of Phytophthora pluvialis in New Zealand compared with Oregon. we found pretty much the same in radiata too. Lower temperatures, lower winter temperatures are a driver for red nettlecast development, or limit red nettlecast development. That's one of the, I guess, the keys to managing the disease is knowing when to spray. And higher mean winter relative humidity favours pluvialis. So mild winters, high humidity, you could well get a disease outbreak. So I'm now going to pass on to Ellen.
Morning, everyone. So here I'm going to talk a little bit about using machine learning to actually model RNC. So the objective here is actually to make use of the machine learning algorithms capabilities to actually understand complex high dimensional data relationships to help us understand whether are there any other external key factors that drive RNC. And ultimately, what we want to do is actually to have a model that is able to forecast RNC severity. And having such a model will actually help us make more informed decisions for disease management. So to do that, we looked at extracting climate data from the NIWA climate station. We also use topographic information such as elevation and slope derived from using remote sensing. And we also use RNC field observation data that we extract from the Forest Health database and site observation. And from these data sets, we actually managed to extract and derive over 130 features that we use to successfully build a spatial climate area-based model for predicting RNC given predicting the severity of RNC for a given location and its associated climate conditions. So to actually go about how we go about understanding what are the key factors that drive RNC, we look at how the model actually uses the features for prediction. So here we first massage and clean the data into a format where the machine can actually analyze, and then we train the model on the data. Once the model is trained, we actually ask the model to generate a feature ranking. So this feature ranking is basically a ranking of how the machine perceives the importance of each feature for predicting RNC. So using the ranking, we then focus on the top 15 important features, and we actually extract the data using those features and correlate them with our RNC field observation. So our feature analysis exercise sort of affirms some of the observations field observations that we observed previously, such as moisture playing a key role in driving RNC. But not only that, we also managed to identify interesting climatic and topographic features that show signs of contributing to driving RNC. So features such as wind speed, distance to coastline, slope, and elevation. We also observe a strong data relationship when correlating historical information back to RNC observations. And this sort of gives us an indication of how we can better model RNC moving forward. Next, we actually sort of test the prediction capabilities of the model using data that we have withheld from the model during training. And here we can see that the model is actually able to predict the seasonal cyclic expression of the disease that we observe in the field from our field studies earlier on. Having said that, we also found out that our model is still not very accurate when forecasting this RNC severity for areas that we have no information about, and the gap between the gap between the accuracy here actually is a strong indication of that we do not have enough information yet to actually have build a generic forecasting model. So moving forward from here, we need actually more high quality information on disease severity. We see that remote sensing actually plays a key role here to actually not only help us gather large-scale information of disease severity, but also more importantly, unbiased and consistent data as compared to if you send few operators out into the field. Yep. So I'll hand it back.
Okay, so Alan's talked about some science that helps us predict when outbreaks are going to occur. So that's, I guess, when to spray or when to control. I want to talk about how to control or what methods we can use. So the first one is copper. Copper is the obvious thing to test to control red needlecast. We've got a spray, operational spray program for Dothostroma needle blight. Wouldn't it be great if we could just transfer all that knowledge and you know, the logistics into a control for a red needlecast program? So we did focus on copper, testing copper to see whether it had the ability to control red needlecast or not. In order to do that, obviously you need to assess its efficacy, and to do that, firstly you need to assess its efficacy, you need to investigate the optimal timing for the application, which is where Alan's work feeds in. And to do that, you really need to assess disease. So we tried four methods to assess disease. One was just assessing trees from the ground, but that's subjective. It has between excessive variation. It's affected by light and weather condition, and now overcast conditions. It's really, really hard to see up in the crowns of very tall trees, and you get obscuring of symptoms through the subcanopy and branches, lower branches on the trees, the target trees, and it's time consuming as well. So what we did is we used an operational spray trial that was established in 2017, just a routine randomised block trial with spray applications in February and November, and a double spray in February and May. And from that, with all blocks, we looked at ground-based visual assessments. We looked at taking colour aerial imagery directly below, or having the target directly below the aircraft or the vehicles, the UAV. We also looked at oblique imagery and hemispherical imagery to look at, I guess, canopy density. And these trees were assessed in September 2018, 20 trees per block, and we looked at the percentage of crown with symptoms. And there was a hint, I guess, that copper reduces red needlecast severity, but it wasn't statistically significant, and there was no effect in spray timing. You'll note that the disease severity was maximum of about 10%, ranged from 8 to 10%, so that's very low. In fact, we didn't get a lot of disease in those trials, and that's normally a fact of life when you're putting in spray trials or trials to control the disease. You don't get disease. It's just what happens. When we looked, though, at red, green, blue aerial imagery with the point directly below, we found different results. So that assessment method was more I guess, sensitive or more accurate in establishing or assessing disease, and we did get some significant results. We found that copper did reduce red needlecast severity using that assessment method. Again, there was no effect in spray timing. So, to conclude, we do have evidence that copper reduces red needlecast severity. There was no effect of spray timing. Aerial imagery was a more effective method for assessing treatment differences than either ground method. but an oblique imagery where you're taking your image on the angle had the best correlation with ground scores, but there were line of sight issues. So really, from those trials and previous ones, I'm confident to recommend that copper application can control red nettlecast. The trial was, I guess, affected by the low disease levels. It would have been fantastic just for that 1 year to have high disease levels so we could determine the effect of treatment differences, but I think we've got enough knowledge now to recommend that copper can control red nettlecast, but obviously there's other methods, there's more long-term sustainable methods, and that's breeding, so I'll just pass on to Natalie now.
In terms of managing disease in the midterm, the obvious strategy here is breeding. We want ultimately be able to deploy resistant lines in those areas that we know are going to get disease. We don't want to be depending on the chemicals that we can use in the short term, but ultimately we want the breeding to be able to deliver the solutions that we want to manage this disease. Again, we all know that there's increasing social licensing issues around using various chemical sprays, and we've got those options for the moment, but really we want to not have those be the only tool in our toolboxes. And the ultimate goal here is that we want to reduce losses, both economic. Obviously, if we can control the disease, we can minimise impacts on growth rates. But if we can minimise the dependence on chemical sprays, that's yet another expense that we don't need to worry about in managing our stands. And then in terms of the work that's been done around Coady dieback, that's ecological losses now that we're talking about as well. Some of the work that's been done in the Healthy Trees, Healthy Future program around screening for resistance to Coady dieback has involved screening seedlings at about age 15 months, and these get placed in these little buckets with inoculum in them, and they get re-exposed once a month. And then this is work done down at Manaki Whenua. They then wait and see which of them develop these characteristic lesions, suggesting that they've been infected. To date, I think the sense has generally been that there's not a lot of resistance that's been found to Coady dieback, but the outcomes from this study have been quite promising. When we look at 40 days post-inoculation, the red bars here, we're starting to see some families dropping off in terms of their survival rates, but it's at this 106 days post-inoculation. Some of these families here are almost completely wiped out. But the exciting thing is that there are some families at this other end of the spectrum that are not completely wiped out. And the fact that this is happening on a family basis, again, is suggesting that there could well be a genetic basis for this tolerance to Coady dieback as well. So this is a really exciting and really promising result. This has then been repeated with collections that have been done in subsequent years. Similar results were seen from the seedlings collected in 2017. The screening for the 2018 collections are still currently underway. This time the strategy was more to actively target within infected stands, to go and target the individuals that seem to be doing much better than the surrounding trees. And from that study, we've also done some preliminary genotyping work as well, trying to see if we can start to uncover some markers that might have correlations with resistance to this disease.